This week, we'll look at a different group of protozoans called the Apicomplexans. Apicomplexans are a group of protozoans that were formerly known as sporozoans. Unlike most of the ones from the last lecture, these protozoans can reproduce both asexually by multiple fission, also known as schizogony, of trophozoites to produce multiple new cells called merozoites, and sexually by differentiation into male and female forms called gametocytes that fertilize to form zygotes. Zygotes encyst, forming oocysts, and the sporozoites that develop within are released to invade new host cells. Pathogenic protozoans include coccidians such as Cryptosporidium, Cyclospora, Toxoplasma, and Plasmodia such as malaria. The first disease we'll look at is Cryptosporidiosis. Some cases are asymptomatic. However, for others, about a week after infection, gastrointestinal problems such as stomach cramps, watery diarrhea, which is often severe, dehydration, nausea, vomiting, fever, and weight loss may occur. Symptoms are self-limiting and resolve after approximately 1 to 2 weeks in healthy individuals. However, in immunocompromised individuals, chronic diarrhea may sometimes become life-threatening. The life cycle of apicomplexans are more complicated than the amoebae in flagellates in the last lecture. There is an environmentally hardy external stage called the oocyst, which when ingested exists in the intestines to release sporozoites, which develop into trophozoites in epithelial cells where they reproduce asexually. Merozoites that are released from merons infect new intestinal epithelial cells. Some of these merozoites develop into gametes that differentiate into male and female forms to reproduce sexually. The fertilized zygotes develop into either thick-walled oocysts, which are passed from the host in fecal matter, or thin-walled oocysts, which remain in the intestinal tract to cause autoinfections. Transmission is via the fecal-oral route, either directly due to poor hand hygiene or indirectly through contaminated food or water. Cryptosporidiosis is prevalent in North America and around the world. Outbreaks have been associated with contaminated drinking water, recreational water, and food. Major cryptosporidiosis outbreaks due to contaminated municipal drinking water include the 1993 outbreak in Milwaukee, Wisconsin, with more than 400,000 cases, and a series of outbreaks in various cities in the Okanagan region of British Columbia in 1996, with 10,000 cases, and more recently, an outbreak in North Battleford, Saskatchewan, with more than 5,000 cases. Before I arrived at Ryerson, I investigated all of these Canadian outbreaks at the BC Centre for Disease Control. In Canada, Cryptosporidium oocysts are commonly found in sewage, raw surface waters, and occasionally in treated water when breaches occur in the filtration system. Humans and other animals, especially cattle, are important reservoirs. There are several risk factors, such as exposure to young, diaper-age infected children or animals, participating in recreational activities such as swimming and inadvertently ingesting contaminated water, hiking or camping, or traveling to endemic areas and drinking unfiltered or inadequately treated surface water or groundwater from shallow, unprotected wells. Sexual contact that results in exposure to fecal matter from infected individuals is also a significant risk factor. In comparison to giardiasis, cryptosporidiosis rates are only a fifth of these rates in the past 10 years. There were peaks in 2014 and 2016 in Toronto, when the majority of the cases were travel-related cases. In 2014, a public health investigation was undertaken, but a common source of infection for the locally acquired cases was not identified. In 2018, the increase in all jurisdictions occurred due to changes in the lab diagnostic test from microscopy to PCR. The last Toronto public health investigation on exposure sources and settings of cryptosporidiosis cases was carried out in 2016. There were 87 cases of cryptosporidiosis in 2016, a 71% increase over the past 5-year average of 51 cases, and the highest incidence rate observed in Toronto since 2006. This was partly driven by increased reports from July to September. The majority of the cases during this period were exposed through travel abroad. Recreational water and swimming pools were the next highest exposure source and setting, although this was in less than 10% of the cases. The lab diagnosis of cryptosporidiosis is by the detection and identification of oocysts in stool specimens. Cryptosporidium oocysts are round, 5 microns in diameter, with 4 sporozoites inside that stain up with acid-fast stain. Molecular methods are used for species-level identification. There are 2 major human pathogenic species, Cryptosporidium hominis, which is mainly a human pathogen, and Cryptosporidium parvum, which is mainly zoonotic, as it can infect both humans and other animals, for example, livestock and wildlife. Other Cryptosporidium species also exist, some host specificity, others are more broad range. The infection is self-limiting and healthy individuals normally recover without treatment. Fluid replacement therapy should be administered to infants to prevent dehydration from diarrhea, and drug treatment, which didn't exist for a long time, is now available for cases older than a year of age. Cryptosporidiosis used to be a life-threatening disease in immunocompromised individuals and those with AIDS. However, antiretrovirals are now so effective that symptoms have decreased to the extent where cryptosporidiosis is no longer a major problem. Oocysts are extremely hardy in the environment and can survive in cold water for as long as 18 months. They are resistant to chlorination and alcohol disinfection. To prevent transmission from infected animals, contact with animal feces, particularly from young animals, should be minimized by wearing disposable gloves when cleaning up. And good hand hygiene is also essential after contact with animals, such as livestock or pets, and their living areas. The same strategies for giardiasis apply in childcare and recreational settings. Basically, children with diarrhea or diagnosed with cryptosporidiosis should not use facilities for at least 2 weeks after symptoms have resolved. Children should have frequent bathroom breaks. Diapers should be checked often, and soiled areas cleaned thoroughly with soap and water after toilet use or diaper changes. Showering is recommended before recreational water use. Inline secondary disinfection systems such as ultraviolet or ozone should be installed to reduce contamination of treated recreational water venues. Ingestion of swimming pool water and untreated water or ice in areas with unsafe water supplies should be avoided. In areas where drinking water safety is questionable, drink bottled water and heat water to a rolling boil for 1 minute before use. Water can also be filtered through filters with absolute pore sizes of 1 micron or smaller, or NSF certified standard 53 or 58 filters. Sexual exposure through oral contact with the anal rectal area or contamination with fecal matter should be avoided. There is presently no provincial or federal legislation on maximum acceptable concentrations in drinking water, but a recommendation that at least a 3-log reduction in protozoan cysts and oocyst levels should be achieved by treatment technologies. Unlike the United States, monitoring is not mandatory or routine in Canada. Detection of oocysts is sometimes carried out during waterborne disease outbreaks. Immunofluorescence staining and differential interference contrast microscopy are used to detect the presence of oocysts concentrated from large volumes of filtered water. The pictures show a fluorescent Cryptosporidium oocyst on the top, and the same oocyst viewed using differential interference contrast microscopy. Even though 1,000 times magnification was used, you can see how small the oocyst is, and it really needs a trained eye to detect and identify this parasite. Waterborne protozoa can be collected by filtration through yarn wound, foam, or membrane cartridges with 1 micron or smaller pore sizes. A diagram of the sampling setup for collecting protozoa is shown here, which can be connected easily to water supplies to facilitate the filtration of large volumes, that is around 50 liters of drinking water, for lab testing. This is a picture of the water sampling device, which has been plumbed in place for collecting Cryptosporidium oocysts in a pump house. This was set up during a cryptosporidiosis outbreak in an agricultural community in British Columbia. The drinking water was only treated by settling in a small outdoor reservoir and then chlorination. Cryptosporidium oocysts were detected in the sample that were collected. a boil water advisory was issued, and the number of cases dropped.